Hi, everybody. Thank you so much for joining us today. I am so thrilled once again to have Dr. Judy Mikovits on. Judy earned a BA in chemistry from the University of Virginia in 1980 and a PhD in biochemistry and molecular biology from George Washington University in 92. Her PhD thesis defense changed the paradigm for therapeutic treatment of HIV. For this work, she was awarded the Graduate Student of the Year in 91. In her 35-year quest to understand and treat chronic diseases, she has co-authored seminal papers culminating at least a decade of research in each of the four fields: immunology, natural products chemistry, epigenetics, and HIV/AIDS drug development in 99, when she directed the Laboratory of Antiviral Drug Mechanisms in developing therapeutics and diagnostics for HIV/AIDS and AIDS-associated malignancies. These are the therapies that are still standard of care 25 years later and credited with saving millions of deaths from HIV/AIDS. In 2006, she became attracted to the plight of families with neuroimmune diseases, including, can you say that?
Myalgic encephalomyelitis.
Thank you. And chronic fatigue syndrome and autism. And she has been primarily responsible for demonstrating the relationship between environmentally acquired immune dysfunction, chronic inflammation, and these diseases. In 2014, she and Kent Lively co-authored the book Plague. And here is a cover of the book, which I highly recommend everybody get. Take it away, Dr. Judy.
Well, thank you, Nancy. It's delightful to be back again, getting a little bit more savvy at giving these talks and these webinars. A few disasters, but doing pretty well. So what I put together for you today is just a talk, and I'm going to go ahead and share the screen now with the slides that I have. And let's see if I can get that up there. So looks like I succeeded at that.
Yes, you did.
Okay, now you should see the first slide, and just stop me if there's questions that need to be addressed, and I'll escape from that full slide format. But at the end of my ordeal for my team and my discovery of the inconvenient truth about the nature of a new family of human retroviruses in many neuroimmune diseases and cancers, the conclusion of the federal government was that the blood supply and the vaccines are safe, and you can all go home now. We, that is the HHH, NIH, FDA, and CDC, debunked that problem, and that problem would be Mikovits and Rossetti. What plague is the story of the scientific establishment's use of the systems of grants, the systems of publication, the system of funding, and most importantly, the media, including the scientific literature, to in fact deny the retroviral etiology of human diseases. And as Frank Rossetti said at that time, and continues to say, like the plague of the Middle Ages, this is politics, it's not science. I have there the plaguethebook.com, markconsultinginc.com, our phone number and email for questions. We'll have those slides up, the slides I show you today, up on both sites, and they'll be freely available, and we try to answer all questions. So
Dr. Judy, we need to see your screen.
Okay, so can you see my screen?
No, we're still seeing you.
You're still seeing me, so let's go back to screen share.
Yeah, we saw you for a second. I mean, we saw the slides for a brief instant.
Okay, well, let's see if this works. I'll start it again. Does that work?
Yeah. Yes. Yep.
Okay. Okay, so I'm on the first one there, and I'm going to just let it go because when I use the presenter format, I just talked about the first one there with the cover of Plague the Book and our emails and what I just said. So basically, the book and in this talk is going to walk through the history of the politics as well as the science of human retroviral disease research. So on the second slide there, you see that in the 70s, there were a number of human retroviruses that were associated with human disease, published and identified and discussed in the literature for a number of years. One called HL23 from the Gallo lab was later shown to be a mixture of 3 primate viruses in histiocytic, it doesn't matter what kind of cells, and shown to be a contaminant. And then they weren't able to reproduce it in other placenta. What Frank shows in this slide, which Frank Rossetti made, is what he says is none of these papers were ever attracted. In fact, the people who publish these papers are in the National Academy of Sciences and still have very large profitable labs within the government. So that investigations don't give full answers in the first papers usually doesn't result in the destruction of scientific careers and lives. But when you're talking about the human retroviruses that we discovered and going against the establishment, that's where the plague of corruption started back in 2009 and continues to this day. So John Coffin and the establishments made the pronouncement, as you see them on TV and Tony Fauci all the time, making these pronouncements that these things don't exist, and anybody that finds anything in cells, they're obviously contaminant. So shortly thereafter, unfortunately for all of humanity, Frank Rossetti and Bernie Poish didn't listen to John Coffin and went on to isolate, I show on the third slide there, this discovery of human T-cell lymphotropic, it was called T-cell leukemia virus first, because only the adult leukemia was known. In 1980, they isolated that. It was endemic to regions of Brazil and Japan, and the majority of individuals never have a symptom from being infected with this virus. There's only a 5% lifetime risk of developing either type of disease. So the adult T-cell leukemia was the patients from whom they initially isolated the virus, and it has a very long latency and develops as the infected individual develops immune deficiencies. That is, the immune system can no longer recognize or kill the leukemic T cell, and that's when the tumor takes over and kills the host very rapidly. Importantly, the inflammatory syndromes that are now firmly associated with HTLV1 weren't realized until a decade later. So there's HTLV1 associated myelopathy, or something that was originally called tropical spastic paraparesis, because these people couldn't walk, they would fall down, they would end up in wheelchairs, as you see on the right-hand side of that slide. The people are standing there in braces. Really importantly, what has been recognized in the next 30 years is that there are, shown in the next slide, there are many factors that are important in the development of the chronic diseases associated with HTLV1. This paper here, published in 2007, describes what they call a subacute progression of the tropical spastic paraparesis. That's almost as bad as myalgic encephalomyelitis, but since I've been doing this 40 years, I can say them both now. So this HTLV1 associated myelopathy, it's a multiple sclerosis type disease. But what they were finding in this paper was that in northeastern Brazil, there was an extremely rapid progression of the disease, putting that infected individual over there on the right in a wheelchair within 2 years. And 8%, and in the studies they looked at, a couple hundred people in this paper, it was all women, and 8% had a very rapid disease progression. In Peru, which is a different environment altogether, 21% had a rapid disease progression. And importantly, there was absolutely no difference in viral load. There was no difference in how much virus was there. So I say this to the clinicians and the practitioners because we all have the need to run off and see and identify the viruses, or identify the proteins or the pathogens involved, the bacteria, and try to understand which is more important in a given situation. And while the virus is necessary and sufficient to get disease, as we see clearly from these rapid progressors, it's not the only thing going on. Hormone differences, environmental differences, heavy metal toxicities, food, genetics, everything plays into the development of disease. And what they found that's the most important for your audience in this discussion today is that early recognition, not of the virus or the potential for the virus infection, is critical. It's the early recognition of the symptoms. Suppress the immune system. It's the overactive immune system and leading to the disease. And if we can suppress the immune system, then we can get those victims out of the wheelchair, slow down the progression, and in fact stop the disease. So it's really not about diagnosing and testing the virus, folks. And I get that question all the time. It's about looking at the immune system, and we can talk, and I'll talk about it a lot more as we go through today and in the future. So in the next slide.
Judy, so the difference between the people in Peru and the others that had a 21% have rapid progression, what do you think is the difference?
Co-infections. One of the studies we did back in the 90s was one we called oncoweed. We thought something in their food was activating the expression of the virus so that you couldn't keep it latent. And oncoweed we did in HIV, and that's exactly what happened. There was a compound in a plant that was indigenous to the northern area, Africa, and those people were dying of AIDS very quickly because that was activating their virus. So it was sending their virus into more infection, even though the copy number was the same in the genome of the peoples throughout all of Africa. It's which factors in the food, in the environment, suppress the ability of the immune system, the methylation machinery, other things from clearing and silencing the virus. So in this case, it probably, I don't know the answer from this group in Peru, but in HIV and in Africa, it was the plants they were eating activating expression of a latent virus.
Okay, thanks.
Which is pretty cool when you think about it, because that's how we think about, and we'll talk a lot about natural products, and because that's why it was important for you to read that short bio, because it was my background in natural products chemistry, which is not Frank's background, that enabled us to form these hypotheses and ask these very important questions all the way back in the 90s. And that's why I put up this next slide, which is political influence on scientific research and the impact it has on us all. So with regard to HIV, which was without, as you said, without the isolation of HTLV1, we would have never known how to find and isolate and understand and study HIV. And so at the time in 82, men were dropping like flies of these rare diseases, of these co-infections, of these tuberculosis, Kaposi sarcoma, things nobody got in the developed world. So along comes in 1991, so the group ACT UP That's where it says silence equals death. They used to go to the scientific meetings we attended and basically stand there with those kinds of placards and say scientists are murderers. And when I was very young, I was only in my 20s at the time, 25, I was just taken aback by this, thinking, to tell you how naive I was at the time, thinking it's your behavior that created this. I'm trying to help you and you're throwing things at me. Well, of course, I've learned, through the 3 decades of research, it's not the patient's behavior any more than it's the virus alone. It's not what you eat. It's not what you do. Does that contribute to it? Of course it does. But in the end, it's the virus dysregulation of the immune system. So we made a population out to be lepers or pariahs and excluded them and threw them away. And that's why all of the politics. And when we wrote Plague the book, I wanted to write it when Kent and I told the story. I wanted Kent to tell it like the band played on. So that is a movie with Alan Alda. And when you walk through that book, you can't possibly not see the rally cry when we first identified this virus in patients, in women and children with this chronic fatigue syndrome. Nothing could be worse. You get up, you're lazy, go do something, eat right, take care of yourself, exercise, and you'll be just fine. So the kinds of abuse that the patients get isn't a whole lot different than the gays in the early 80s got with this, when HIV was identified to be causing disease in this patient population. So the band played on. There were many, many deaths before the establishment believed, it's not religion, it's science, in a retroviral cause. And of course, you can have HIV and not have AIDS. And that's why Magic Johnson is in the middle of that picture, because that PhD research thesis that you talked about in my bio was defended 1 week after Magic Johnson was declared to have been infected with HIV. And the only question that thesis committee asked was, based on your data, based on your work, will he or will he not die of AIDS? And I said, if he changes the paradigm and he doesn't listen, to those who are saying, oh, don't take these highly toxic antiretroviral therapy and don't start now, wait till your immune system falls apart. So he had to be brave enough and courageous enough to stand up there and say, okay, I'm going to trust you. This research is new, not me in particular. It was David Ho and company. And I'm sure that nobody would ever know this was my PhD thesis defense, because of course the literature published it their own way and they got to rediscover it themselves. And the men do this in science, and they continue to do this to this day. And that's why Plague the book's written that way. But we changed the paradigm. We changed the way we thought about it. We changed the cells that were infected. Funny enough, that work was called contamination, because I was an idiot. What people said before that, you idiot, don't you know only CD4 positive T cells are infected? Your preparations must be contaminated. Well, of course they weren't. And of course I was right. And he's still alive 25 years later, and I'm still right. And I'm just delighted. I hope one day I can meet him, because without his courage, we wouldn't have changed the paradigm. Somebody has to step up there and say, you know what? I'm not going to change anything any other way, and I'm going to die anyway, in all probability. Let me try. And he tried what were thought to be so toxic and so dangerous, and he's just fine. And he also had a healthy family after that point as well. So we can even have healthy children if we're infected. So the next slide shows you the chronic diseases that are associated with human retroviruses. This morning I updated this list, and I could update it even more. But I wanted your clinicians to see, because just like we want those clinicians in Brazil and Peru to understand when somebody hits a wheelchair very rapidly, And there's no evidence of infection of anything, because most of the time you can't just go looking for all of the viruses. And you're looking at some of these diseases, think retrovirus, think immune suppression with either targeted molecules, or even steroids aren't a good idea in some cases, but this is how it was the combination of steroids and antiretroviral therapy, which Magic Johnson is still on to this day, that actually kept the disease from progressing. So when we see these diseases, how much more multiple sclerosis are we seeing in our world? How much fibromyalgia, chronic Lyme disease, which is clinically indistinguishable from ME/CFS. And there's an entire chapter in Plague about the fact of we found evidence of the retroviral activity in virtually 100% of those patients who wouldn't recover from the chronic Lyme, from the Lyme that the Borrelia couldn't be treated and cleared by the antibiotics. And that's because something else is going on that's crippling the immune system. So it always comes back. Well, you can't live your life on antibiotics. That's why we have an immune system. So I'll leave that, that slide up, because all of the diseases of our time, and they're going up exponentially as we add these environmental toxins, and primarily the environmental toxin that is causing the most of this is this ungodly vaccine schedule of contaminants we're injecting, including retroviruses. And we'll go on from there. So I just show you one example of lots and lots of literature in ALS, Lou Gehrig's.
Judy, can I ask you a quick question?
Sure.
With Magic Johnson, was he also given a specific diet to help him?
I actually don't know that. I'm sure that I know that he's still treated by a physician in David Ho in New York. I know he still has a team of doctors. I actually tried to contact him recently and just asked those very questions. But he's pretty protected by those doctors. As I say, I hope one day we can establish communication when I'm no longer considered persona non grata in the scientific establishment, which hopefully will occur sooner than later.
So I also want to ask you about Lyme. So what kind of, you're saying that the treatment that people are receiving now does not give any kind of permanent relief. Did I get that right?
Well, when we use the antibiotics for the Borrelia and for the other co-infections, Babesia, Bartonella, yes, they provide a temporary relief. Same thing with the herpes viruses. We often see herpes viruses co-activated. EBV, CMV, HHV6, lots of herpes viruses. And that's what we saw in HIV/AIDS. So in the book, The Band Played On, and what you're going to see, and what I don't have time to go into, is if we didn't treat the tuberculosis, if we didn't treat the Kaposi sarcoma, if we didn't treat the mycoplasma pneumonia, those patients would die. So you had to treat things quickly. And you have to stop both families of pathogens, from just doing any more damage to the immune system. And that's where the physicians, and we work with physicians, this is exactly what we do. We see patients where there's a retroviral, suspicion of a retroviral role in the disease. And I'll have the doctor say, okay, this one, which would you do first? The acyclovir, the Famvir, the antimycotic, antimold, and then it's a matter of a clinical history and really personalized medicine. But you have to treat with the antimicrobials, but the disease will always go back. And it's about 6% of those diagnosed with the chronic Lyme disease.
Okay. So is it possible to treat these kind of problems using just natural therapies?
Yes and no. And I will talk about that at the end. I have a few natural therapies, but yes and no. At the point where you're so clinically ill if you don't come in with a known reverse transcriptase inhibitor, if you don't come in with a known liver detox, if you don't come in with the antimycotic, if it's mycoplasma, you lose the patient from mycoplasma pneumonia. So yes, nutrition and natural products. I mean, natural products are the drugs we developed. And I'm not sharing that screen right now, so you can't see my hands waving. And that's what we did. So we isolated from those very trees, from those very things. They are the start of what were those drugs. And we're learning about what we used to call off-target effects or bystander effects of those drugs. So we could, as I talked several years ago, we're teaching old drugs to do new tricks in new diseases. And that's why it's very much, it's not a single drug. It's not a single anything. I'm not sitting here saying everybody run out and take highly active antiretroviral therapy, because we pick and choose these therapies very carefully. And most of them were not developed as HIV drugs, as antiretrovirals. They were developed for cancers and parasitic diseases, narcolepsy, actually tropical diseases, much like tropical spastic paraparesis. They were developed in a disease where you didn't know the virus existed. We didn't know Kaposi sarcoma was a herpes virus until we found it from HIV patients. So you have to kick the immune system down so far to find these viruses. That's why we don't recommend looking for them, because you're usually going to get, no, they're not there, because your immune system is good at clearing and silencing these pathogens. That's what it's there for.
Thank you. I'm sorry to take you off track here, but
it's actually part of track, and that's why I don't have all that many slides in here. I mean, it might seem like a lot to you, but anyway, so this is just one example in Lou Gehrig's disease. So, Lou Gehrig's disease can be called motor neuron disease, can be called multiple system atrophy, can be called Lyme disease, can be called a lot of different things, because it presents itself in a lot of different ways, initially only as fasciculations or shaking or weakening of the muscles and things like that. So as I said, with the tropical spastic paraparesis, recognizing these things early and thinking about the big picture and no closed mind is how we can help the patients the best. So I just show this primer, and again, we'll provide these slides to anybody, of the role of human retroviruses in chronic disease, because there are a lot of cancer causing, shown on the little tree. They gain leaves, they make more cells. And on the other side, HIV kills the cells, and it's the cell death that's killing the patient. So their RNA genomes, and first they must integrate into your DNA, and the DNA of the host of the cell. And it's that cell where they live that they can do the damage and be carried on for generations. So for instance, if you infect a B cell, then you're going to develop B cell diseases like multiple myeloma, like chronic lymphocytic leukemia, like mantle cell lymphoma. These are all of the lymphomas I'll show you later that we found evidence of these viruses in, and they were in that original table. Can you have these viruses and not get those cancers? Of course, that's what we're talking about. But it's important to notice that as much as 15% of the human genome is these endogenous viruses. And that's over the millennia, your immune system cripples them, so they can't be expressed. Well, when you cripple your immune system, they become expressed. And then whether either with environment, with bad food, with GMOs, with other co-infections, then your endogenous elements become expressed. And as we'll see later on in great detail, they can recombine. They can create new things, and they can recombine and create these infectious viruses, much like the origin of XMRV into the human population was introduced as a recombinant, which came out of laboratories into the blood supplies and into vaccines. So the pathways for the physicians and clinicians in the audience, healthcare professionals, the pathways, the quote at the top comes from a article that Hillary Johnson wrote in February of 2013 about chasing the so-called shadow virus. And I put that in there because, of course, all of these things look like retroviral diseases. It wasn't rocket science on our behalf to look. It's what we did. They look like acquired immune deficiencies. There's inflammation. There's dysregulation of hormones. There's highly elevated reactive oxygen and reactive nitrogen species. There's immune deficiency. That means you have, let's just say, I think we said in the last talk that you have B cell subset, antibody subset deficiencies, IG, and so doctors that are using IVIG to replace what you're missing are putting a Band-Aid on it, and a dangerous Band-Aid on it, because you're not realizing that you're taking out a component of your cells, and you're further dysregulating your immune system when you don't consider a retroviral elicited pathogenesis. Insertional mutagenesis means that virus, when it integrates, sits in front of a gene, at the promoter of a gene. It's the on and on switch, and tells it to go on when it shouldn't or off when it shouldn't. And that's how they create disease. That's 40 years worth of study. And these pathways are key. Epigenetics, DNA methylation. We all know about MTHFR. You start thinking about the pathways that silence and control retrovirus, and you start to understand the diseases, and of course then you find the elephant in the room, that the retroviruses are in fact involved with all the diseases that are chronic diseases of our age. So the appearance in the chronic fatigue syndrome and autism in the medical literature is, I believe, chapter 5 of the book. It doesn't matter. There's the chapter title there. That first outbreak was in Los Angeles in 1934 and 35. It was the child was given this yellow fever vaccine. I think it was a doctor's child. This is a fascinating chapter. If you buy the book only for that chapter, you should, because it's a bonus, because I didn't have anything to do with writing it, so I'm not long-winded. This was all Kentek and Lively's research and brilliance. I just made sure it was scientifically correct. But the first cases were among those parents with a medical degree or scientific backgrounds, which starts to give you clues. Dr. Byron Hyde, who had been doing research into CFS, gave the quote from an interview he had with Kent in 2013. And the original document from the study of that outbreak in Los Angeles said it was caused by this immunization, and it was the transfer of the human infectious material. In fact, so it was a great experiment. You cause a disease with the transfer of this material. But they had a huge fight with the head of the public health system, and it went on for 6 months. So this guy can't get his papers published. And finally, it was agreed that in order to publish it, they had to take out anything that had to do with immunization, because that would set back all Americans, and which is hogwash. And of course, now we see all this disease that's a disaster in our country because of vaccine injuries in more than 20 million people by the spread of these infectious agents through vaccines and a contaminated blood supply. So the next slide shows you the search for retroviruses. This just shows you I'm not the first. I wasn't the first. Other people lost their lives economically, scientifically, and actually. This was the paper by Elaine de Freitas that was published in 1991. I made that very, very large. So just when we discovered what we discovered about Magic Johnson and other subsets and changed the paradigm, here was another similar virus in these patients with chronic fatigue immune dysfunction syndrome, which at that time, by Shai-Xing Lo and others in the federal government who were working on this, it was called non-HIV AIDS. It was women, it was children, it was a lot of people. There was no evidence of HIV, but just like with HAM TSP, they were clearly with profound disease, and it is from that, which I'll show you later. I've got a slide out of order, but at any rate, it was from those data that Elaine DeFreitas claimed a retrovirus, the corrupt, the cesspool of disease, of deception and corruption, which we call the Centers for Disease Control, destroyed Elaine DeFreitas's life, and they said, oh, we cannot reproduce the data, and it was roundly dismissed, only to cost another generation of people their very lives, and caught countless years of suffering. So the next slide shows you that in 2000.
Judy, can I ask you a quick question?
Sure.
Yeah, give you a chance to come up for air. So I have a few questions from our listeners. One is, doctors, is it recommended to use antiretrovirus therapies on patients diagnosed with ME/CFS within a year of onset?
Yeah.
Okay. That was an easy question. And then,
in fact, to answer that, when our paper got published, I said my first thought and my first comment was, now we don't have to wait 6 months without ever knowing what's causing this to get a treatment and solve the problem. Right. And that, of course, was not what happened because of the politics.
Right. So the sooner the treatment, the better the outcome.
Absolutely. As soon as you see it, clinicians.
Okay. So I have another question or a statement from Jamie. This is me. I have multiple autoimmune diseases and primary immune deficiency. My completely unvaxxed son has autism. How do we get tested for these?
Well, as I said to Jamie, you don't want to test for the viruses. You want to, because of course they're not going to come up because they've taken them out of the databases and they've killed everyone that tries to even dare go near there. So what you want to do is fix all of the autoimmunity, and we can do that. We have a lot of therapy, so I can, if Jamie wants to drop me a note, I can help the son. The unvaccinated is her son's diseases and genetic susceptibilities were probably largely responsible, and we all live in this toxic soup that is GMOs and heavy metals and everything else. So that they're totally unvaccinated is not surprising, but at any rate, I'd happy to talk and answer that question, because we've done so much in immune therapy. We can do a lot to help both.
Okay, great. Thank you.
So I just show here that in 2007, by one of the new sequencing technologies, Bob Silverman and Ron DeRisi identified sequences that were later they cloned in the laboratory and called this retrovirus. Well, they cloned it from pieces and parts, and we didn't find out any of that. That's a whole chapter of the book. What mattered is the kind of virus it was, and that they integrate preferentially into CPG islands. That's the promoters of genes. That means this gamma retrovirus, which has never been found in humans before, and he identified sequences. And of course, the next slide shows that we isolated the virus first. The first isolation of the murine leukemia virus-related retroviruses from humans was, in fact, our work, the publication in Science. But 4 months before that publication in Science, chapter 8 of our book was the invitation-only July 22nd meeting. This was every head of every level of the government, and they listened to every bit of my data, so much of the immunology, so much of the children, women, cancers. None of it was in the paper because the journal suppressed it and cut it out of the paper. But here was John Coffin's comment, his exact quote: "You mean all those sequences we saw were real?" So the sequences that Silverman and DeRisi found in the patients, they were only a couple of hundred nucleotides long. We isolated the virus of what they were, but the clone sequence didn't turn out to be the sequence that we isolated. And that was where all the confusion in the testing came and everything else. No matter, the sequences are real. There are many families. 1 month after our science paper came out, you see on the next slide here, Gary Owens came to a meeting at the Cleveland Clinic that I attended, and he put up on his slide what he called XMRV2, and he showed you how similar it was to XMRV1. And for those that like phylogenetic trees, it had a 93% overall homology, but it was clearly quite distinct from what we had isolated. He dared call it XMRV2, at which time John Coffin said, "You can't call it that. You can't, you know nothing about phylogeny. You can't name it anything. You listen to me." You know, it's just this, I really hope I live to see the day that John Coffin goes to federal prison, because he belongs there with Tony Fauci. At any rate, in the next 6 months, the Lasker Award winner Harvey Alter and Shai-Xing Lo. Shai-Xing Lo worked for the FDA at that time, and he had kept a freezer of samples that he never thawed out. And when we published our sequence and our work and the virus and the proteins, he came up and he did the PCR, and you see there the paper that eventually got published in PNAS, and you see his quote from about 6 months earlier. He said there was a dramatic association. It was 86% and 8% in controls. Now we're approaching 6.6% in controls. Now we're approaching 20 million Americans that are infected, that are walking around, like I showed you with HTLV1, that are ticking time bombs. If only 10% or 15% get disease, what is the next thing that's going to kick them over the edge? Of course, they were forced in the plague of corruption to retract their work, which they should never have done, because in fact you can't reproduce work unless you use the same samples or the same tissue samples, and that's a whole chapter in the book on the Lipkin fraud, which we'll talk about a little bit later. The phylogenetic tree, when we went back, we shared both Gary Owens, every one of our samples with Gary Owens. We shared every one of our samples with Dr. Lowe and Alter, and they shared theirs with us. We used our tests and got the same answers. And in fact, we found Gary Owens' virus in a number of our samples that we had called negative, to the point where when you did the phylogenetic analysis here, you can see the huge range in our original cohort. And this person down here that I'm highlighting is close to a mink cell focus forming virus, and she had a very aggressive disease. And she was in fact one of the ones successfully treated with antiretrovirals, but not before she was treated for the herpes viruses, because of the level of EBV in her system was causing her more immune dysfunction. But interestingly enough, this was from our original cohort. We were essentially 100% positive when we looked at all the family members. So shortly after Harvey Alter's paper came out, and after we appeared at the first and last XMRV workshop, which is another chapter in our book, I'll look and tell you which one. It was called, let's see, chapter 16. So the chapter about Harvey Alter is chapter 14. Chapter 6 is the first and last international workshop on XMRV. And at this point, after we showed evidence in a group in London by 3 different assays of many, many strains, a group in Ireland, a group of patients, we had been working with cohorts around the world, and the answer was the same. And we showed those, even though they tried to keep us out of that meeting. And Harvey Alter at the first and last workshop, not Harvey Alter, Francis Collins asked me where we got the controls of 4% from the London study, and I said, the blood supply. So he immediately commissioned Tony Fauci, the head of the NIAID, Allergy and Infectious Disease, and Ian Lipkin to oversee a study to end the debate once and for all. 4 days after they convened that meeting, Suzanne Vernon, a longtime CDC employee who became an advocacy, MECFS advocacy organization head, came out with this blog and said, agency heads are worried, scared to death if XMRV works out. Because, of course, now you've got worse than HIV/AIDS, which at the height in the United States was 1 million Americans. And they were many infected by contaminated blood supply. Now you've got 20 million Americans that were infected by contaminated vaccines and blood supply. And they've got a real problem. Additional proof, another laboratory, Sidney Grossberg, came up and had isolated this virus in this cell line. He developed a cell line and isolated the virus from a patient from Wisconsin. I've talked to him since. It was a 30 plus 34-year-old woman. She eventually recovered, but at the time he isolated the virus, she was bedridden. He never knew the sequence. He only had a picture. An electron micrograph. He only knew that it was a gamma retrovirus. And when he used the appropriate consensus PCR to detect all family members, he came up with exactly what we came up with, a gamma retrovirus that looks like MLV, and that sequence is largely unknown, but the gag and the envelope are 100% what we showed. We know he validated our research. So I'm showing now the next slide, the contamination of the blood supply, the additional strain, and the disease association. So this is where I show ITP, CLL. Idiopathic thrombocytopenia is listed as a side effect, a rare but significant side effect with MMR. Mumps, measles, and rubella. I did a study with James Bucell, the expert on ITP, and we found 30% of patients with ITP were in fact infected with an XMRV. And I say an XMRV because I'm not going to say what the sequence or the strain is, but proteins and sequences and antibodies were detected in those patients. In the inset here, I show a family where a male child in his teenage years died of lymphoma. The mother had MECFS, and the grandmother died of a lymphoma or had a lymphoma at one point in her life decades back. And XMRVs, because they can infect all cells, including the sperm and the egg, in fact, can be carried down throughout generations. And you see at the arrow here, this was one of the patients in our original study. So when we expanded and when one of the positive in our original study with MECFS and XMRV, we found in fact that the cousin who, died literally when he was in the same location she was when she developed disease, he developed the aggressive lymphoma. So we don't understand why we can't get that sample anymore, but because they threw it away at UCLA. So they buried the evidence, so they couldn't figure all of this out like they tried to do and continue to try to do with us. So the blood supply, the next slide shows you, this was a talk I gave March 29, 2011. The conclusions are in the upper right-hand corner. And it just basically shows you the conclusions that the blood supply is in fact contaminated. In that talk, I showed that the serous intercept system would in fact clean up the blood supply. And interestingly enough, 3 years after my termination, arrest, and false arrest, and jailing, and removal from science, shall we say, and society, they approved this test for cleaning up the blood supply, and they approved it to clean it up in ITP. and they approved, but they did, and leukemias, but they did not approve it to clean it up in ME/CFS, where there's just as much, if not more, and in autism. So they sentenced innocent people to lives of suffering and ruin, and they could have been cleaning it up all along, just like I showed them in 2011. Interestingly enough, a lot of the people like Mike Bush, Ian Lipkin, and others are profiting quite greatly at from the money that came from the Intercept, use of the Intercept system. So here in 2 months, or 2 months after this talk, or a month after I gave the blood supply is contaminated, we went to the State of the Knowledge workshop in ME/CFS. And here is where John, after we showed all of the data that I just showed you to show you how many strains there were, they said, that doesn't mean, he said, that doesn't mean there's not another gamma retrovirus. John Coffin says, I think there's enough evidence that's been presented that there are other infectious retroviruses there. The studies will go on. Of course they will. They shut us down a couple months later when we presented the ITP. And then in his last question of the book said, you know, whether the plague feared by Coffin and Stoy had already arrived, but we don't recognize it. Well, we did recognize it, and that's where Frank Rossetti said they see what they want to see, and that's the real plague. So the name game, and the next slide shows you the name game. It began within, you know, a month, as I said, when John Coffin told Gary Owens he couldn't call that virus. He isolated XMRV2, and he isolated it from people with cardiac disease, by the way. So think real hard when you're looking at these cardiac Lyme association and other pathogens. But anyway, they started right away. This is a paper by Adi Gazdar in the summer of 2011. And what Adi is famous for is his solid research on the role of SV40, which came from vaccines and causing great lung damage and damage in people. So Adi, longtime friend of Frank Rossetti, calls us up and he tells us about this, and he has no idea, and he does know, of course, what we're going through. So at any rate, this was the paper that many cell lines are in fact contaminated, including the 60 cell line screen, which I used my entire life to develop cancer drugs. So in fact, when I said that many of the drugs we developed would to treat these cancers would in fact help some of these diseases, it's because they were probably targeting the immune dysfunction that the retrovirus was creating in the cell line. They were certainly, the viruses are certainly adding to the immortalization, that is the ability of these cells to grow by themselves. So in fact, the next slide shows that John Coffin himself published this paper a couple of years later, November 2013. So you see how the media is covering it all up. So they're getting lots of money from the public and they're covering it all up. They've destroyed us. They've taken us out of research, and they're as fast as they can publishing all this stuff to make you think you're safe and you can all go home now. But in fact, replication competent retroviruses can occur in only 10 days in the lab. And when I take blood from you and I isolate various white blood cells and I culture them in the lab and I use these cell lines and I manufacture proteins and drugs and vaccines in these cell lines, in fact, I do it a lot more than 10 days. I usually keep a cell line in the laboratory 6 weeks. This result says, you know, and they said, oh, you can go home now because they don't have the exact sequence of VP62. You see, that's what I was hung on in the end, that it wasn't the exact sequence of VP62, and that's what Silverman said it was when his lab sequenced the isolates we sent to him. So this is how John Coffin thinks we're all perfectly stupid, and we don't realize that, you know, it doesn't have to be the same sequence. Are you kidding me? How many strains and sequences of HTLV and HIVs do we have? Many of which don't cause anything that we in fact recognize yet. So whether we fail to see the clever virus, which does not kill its host, but has learned to live with it. When a disease takes so much from a patient and stops just short of death, this is ME/CFS. These people are in dark rooms, can't hear, can't see, in silence, skin, nothing can touch them. They're in a state of hibernation, as the data just verified from Bob Navio's group last week in the metabolome. Their metabolism and metabolites are such that it shuts down to save every bit of energy so that you don't die. It's just incredible. So the cell lines are all contaminated. The new technology shown on the next slide reveal this. They've gone on to do this. This was a 2013 or 14 publication. They've gone on to use the new technologies and find out, in fact, everything we said was real and so much more. And yet this isn't what you hear about in the news today. And you don't hear about, oh, the blood supply was cleaned up from Ebola and that big outbreak we had a couple of years ago. Well, the blood supply, the Seris technology can clean up Zika too, because it's the same family member as West Nile virus. So in fact, everything that we're saying, they don't need to spend a nickel on Zika. And right now, they just 2 weeks ago, they just gave several companies, the FDA, $20 million to test the blood supply. So what are they going to tell us? They're going to tell us, oh, you can all go home now, you're safe. We didn't find any Zika, because they're only looking for a teeny little piece and a teeny little part. And in fact, they're recombining. And why are you doing that $30 million of taxpayer money anyway, when the Seris technology will clean it up? And we know that. So this slide shows you in a conference call that Ian Lipkin had at the in 2013, a year after the so-called Lipkin study ended everything because he used the wrong patient population in 3 out of 4 cohorts. Well, in that fourth cohort that we managed to slip past Suzanne Vernon, who was controlling the patient selection in that fraudulent study that I talked about from her slide, how they were worried and how they were going to make sure it went away. Ian Lipkin said, in Montoya's cohort, we found 85% in the sample pools. Yep, that's because I picked them. That's because I knew who they were. That's because they were working with me. That's because I showed you the evidence. And in fact, the whole families were infected. And this was the reason he gave. Oh, but the whole family's got them, so we don't need to worry with that. No, we do need to worry with it, because those are the family members that are going to get sick down the line when they're exposed to heavy metal toxicities, contaminated water, vaccines. Even as adults, folks, the cancers are going to come there. The vaccine damage isn't limited to the kids. And this is what your issue is. So in the scramble of the media to clean it up, the WHO, the World Health Organization, gets involved and publishes this paper on the right-hand side talking about adventitious agents in viral vaccines. What they mean is retroviruses, a little SV40, other things. And they're doing four case studies. And what their findings are is they're finding a lot of reverse transcriptases, folks. And those reverse transcriptases are all widely distributed and evolutionarily related. So again, the very enzyme that makes a retrovirus, and they look like avian retroviruses, and they don't have the unknown safety implication. So what the bottom line of this paper is we have to do a better job of covering this up, folks. We have to do a better job of marketing to folks so they don't recognize not only do they not need these vaccines against pathogens that aren't risks to certain populations, but in fact they don't need to be infected by these adventitious agents, which down the line will put them in the medical community probably with cancers and diseases, getting these diseases for the rest of these drugs for the rest of their lives, and lining the pockets of big pharma. So I put this slide in here because this was eventually the study that Gary Owens got to publish, and I mean was permitted to publish. You know, John Coffin's lab worker got in on it there and boosted up their own career, because in fact, the first author, who was the postdoc that worked with us, needed to get her MD PhD. So don't think that the press doesn't control how everything is put out. So in this paper, this wasn't called XMRV2, it was called V4RV, so the smart patients wouldn't be able to Google it. But what they found is the envelope protein alone, that part of the virus that we identified in every single case with reagents that detected all family members, that protein alone could induce vasculitis. All of these diseases have a component of vasculitis, from kidney diseases to lymphoma to the brain diseases to the stroke. Everything you clinicians are seeing, the vasculitis, it's extremely potent at causing that envelope protein alone. And that, in fact, you don't need infectious virus. So when you hear the government say these virus-like particle containing new vaccines, which actually contain retrovirus elements in order to make them form into a virus-like particle, they say you're all safe, you can go home because they're not infectious. Well, we showed in HIV back in the 90s, we showed in HTLV1 neurological disease, and here Gary Owen shows you don't need infectious virus, folks. All you need are these proteins, all you need are these sequences to be expressed to dysregulate the immune system. So all the way back to 1953, the vaccine contamination risk from mouse tissue in humans was recognized in 1953, 5 years before I was born. Most of you still of the age where you're getting sick when they recognized it all the way back then. In our 60s, how many of us are dying very much prematurely of cancers, accelerated diseases of aging? That's what these are doing to our immune system. So they may be latent in mice, but we picked them up, and they can be enhanced neurotropic properties and serious reactions involving the central nervous system. Well, did they end it? No, they continue today. This is how many new diseases are we creating and how many new strains. John Coffin wrote an article to accompany our science paper where he did this cutesy, one new virus, how many new diseases? And what the long and the short of all of it is, how many new viruses are we creating in our laboratories with these technologies, and how many new diseases? This mysterious disease in China and in Taiwan, in different areas of China and Japan, is called by the affected and the infected HIV with wings, because they're getting infected. They get us with not even sexual contacts, kiss, and we knew the aerosolization that Ardigastar found, that it goes through the airs. And here you go. So here it's published in 2011, new Japanese encephalitis vaccines, alternative to production in mouse brain. Are you kidding me? They knew all of this and they never stopped making them, and they never stopped injecting them. I'll give you the literature and the back literature on every one of these. They're on the CDC list. They contain, they literally contain ground up mouse proteins and sequences, those very proteins that could cause the disease. And there's a whole group of young men, young women, children with extremely aggressive AIDS-like, ME-like diseases that we had done work with Sam Chow at UCLA in 2010. And found absolutely there was evidence of XMRV. So they were in the people. We never got to finish that work because of my untimely dismissal from science. But the fact that they knew beyond 2000 and beyond 1953, the fact that in when we talk about xenotransplantation, that's putting foreign animal cells and material into other animals, can create new viruses, and that it's impossible to remove these from the methods we have or had at the time. So, and they knew this in 1995. That the implanting an organ with a dormant endogenous retrovirus in the patient is equivalent to injecting the patient with a live virus. And that's what they're doing, and so the xenotransplantation. Threats masquerading as cures. And I'm going to keep saying this because, and this is what the media is doing today with this whole Zika fraud. This Zika fraud is pure fraud. It's the same family member as West Nile virus. It's been around just as long as Ebola, just as long as HTLV, HIV, in the monkeys, in the animals, and it doesn't do anything until it recombines or in northeastern Brazil. Isn't that interesting? Where are they getting the ones with microcephaly? Northeastern Brazil. Oh, didn't I just tell you that's where we're finding all those TSP-like diseases? So these viruses talk to each other. So here they said how dangerous it is for the donor organ. And I write, isn't injecting babies and children with mouse viruses capable of infecting human cells the same thing? And yet we're being mandated. And yet we're seeing the injury and we're seeing the disease, not only in the little kids, but the diseases, associated with environmental change, with zoonosis, with jumping out of one animal, one species into another. And so here in 2015, they had a draft guidance for industry, which allowed them to have these little particles in them, the FDA. You could make your vaccines dirty because there was no way to clean them up. So in 2015, they withdrew this draft guidance, which said to take precaution to reduce the risk of transmission to the intimate contacts, not just the recipients of the vaccine, but the intimate contacts. And they withdrew that. Why? For fear of public panic? Well, the current vaccine schedule is compounds the damage in vulnerable populations. People, we now know from the movie Vaxxed, and please, every physician, every clinician, every person needs to see this. If you think for 1 minute where the admission of fraud in the paper that is used as the primary paper to this day, that said MMR is safe. 2002, that paper came out, 2002. Just when they had their little guidance that said, and then they withdrew it. And so that Thompson and CDC employees can admit fraud, and nothing has happened since it was published in 2014. Not one congressional hearing. He's still sitting there taking, as are all the other involved conspirators, Ian Lipkin, Tony Fauci, etc., still collecting their $250,000 and then some salaries, and still injecting, and in fact mandating by law that our kids and our families are injected, our families, a flu shot, DTP, every one of them is contaminated, folks, or carries the risk of being contaminated. So everyone is like Russian roulette and putting a gun to your head. So just with HIV, AIDS, and Magic Johnson, activation of the cellular immune system is important in the pathogenesis of disease. So if you don't need a vaccine loaded with aluminum that's designed to activate the very cells that are going to start replicating and recombining the pieces and parts of these viruses that you've been exposed to for a lifetime, and wait for the day you get the perfect storm. So we do vaccinate AIDS patients, and we do it in the presence of antiretroviral therapy, that those reverse transcriptase can't be expressed, that those proteins can't be formed. There is a safe way to do this, and it's time we did it. The toxic components exacerbate the immune dysfunction. I didn't show my favorite CDC excipient table 2, where you see the vero monkey kidney cells, which themselves have monkey retroviruses in them, and they know it. They put it right there on the paper. But this is for our common good. This is for our greater good. They know it. Fine. You have no choice. Inject your child with enough aluminum to cause Alzheimer's disease when they're 2 months old. It's just incredible. So the conclusions are that all retrovirus can and have been shown to infect the cells like HTLV, like HIV. They're passed through milk and other body fluids. And they're quite stable in urine, folks. They're quite stable in urine. And in animal feces and in the GMOs we're using are retroviral backbones. The gene therapy we were using until very recently was in a gamma retrovirus, and it caused cancer in 30% of the kids treated, and they had to disband that gene therapy program. That was 2002. The blood supply is contaminated. Some of the food supplies are contaminated. We have to think about not using raw milk or making sure you use food and things from places like tropical traditions where, you know, we've just, Stephanie Seneff and colleagues have just identified that in fact glyphosate is also contaminating these vaccines as well, because you can't get it out of the laboratories, you can't get it out of these ingredients that contain animal products, and this is exactly what we're showing. So the only slide that is the therapeutic opportunities in the midst of the plague shows you three different reports, recent scientific reports. One that we published in the lower left-hand corner in the early 90s on bakelin. Bakelin is a natural product from Scutellaria bakelinus georgii. Another one showing that the bakelin has antiviral activity against dengue virus. So that's another relative, a family member of Zika, folks. Do we need to spray DEET all over Miami? Nope. All we need is a few good natural products to get all of these RNA viruses. So there are many flavonoids and various things that will inhibit reverse transcriptase. We at the Natural Products Division and Lab of Antiviral Mechanisms back in the 90s, our colleagues have done lots and lots of work. We're working with companies to this day, happy to identify for you clean products and solid products. It's really important to know what's a clean product, because just like the benefit of these, if they're contaminated with arsenic, will do exactly the opposite. And the heavy metal arsenic is a demethylating agent that we use in tumor therapy. So, and that's often many of these natural products actually are detox alone. I made a joke that wasn't very funny a couple of months ago, but it's that all they have to do is throw cannabis into the Flint water supply, and you'll pull out all the lead, and you'll pull out every other toxin and clean the whole thing up and save the people. But of course, Barack Obama says cannabis has no medical benefit. But that said, the stalks in different parts of these plants are different, and they can be just as toxic. Strychnine is a natural product, and it can kill you. So you have to be careful. You have to know you've got clean products. We'll help you know that. We work with different folks around the world, and there are a lot of good companies making good products and recognizing, like Tropical Traditions, how to clean up the food and clean up the natural products, and we can treat it with these things and have great benefit without a lot of toxic side effects.
Well, I have a lot of questions for you. Yeah. Now that I got brain parts all over the walls of my room here. You blew my mind on so many levels. So first question is, are all vaccines contaminated?
All vaccines are contaminated with something. So whether it be glyphosate, whether it be Vero monkey kidney cells, whether it be histidine, which is going to throw off your mast cells. Yes, all vaccines. And as far as retroviruses, you know, we asked for almost a decade, we asked to get samples of vaccines, and you know how far that goes. All you have to do is cross-link, is put a vaccine in a microwave for 30 seconds, and I won't be able to find any sequences in there because of the way PCR works. So yes, all vaccines are potentially contaminated. So the point of the matter is, if you get 83 shots before you're 18 years old, and I found 30% of them are contaminated with gamma retroviruses, what are your chances? And how much, and at what point, so.
So we also had some questions here. How do we bring this up to a doctor to get them to look into it? If you're having well, let me go back. The first question she asked, this is from Gloria, is an organic non-GMO diet important? And what can my 33-year-old son take to reverse these illnesses? He is now having lupus symptoms, and yesterday his lungs started to hurt. My guess is you got to go organic whether you're sick or not.
Oh yeah, you have to go organic whether you're sick or not. All those healthy carriers out there are time bombs, because it's not only one thing. That's why I showed so clearly they don't get HTLV1 doesn't cause anything other than adult T-cell leukemia, but only 1% of the people with HTLV1 get adult T-cell leukemia. EBV doesn't cause cancer, but a very small 1% of EBV-infected people get Burkitt's lymphoma, a cancer firmly associated with EBV. So yes, you have to go non-GMO organic. And that's where we get into the socioeconomic, because you basically you're basically killing off the people who can't afford to eat that way, and it. So yes, you have to go non-GMO organic. There are some really good natural products that not too much, a couple $100 a month could make a huge difference in the life in a supplement, but you have to choose it wisely, and it's a personal decision. You just cannot walk into GNC. You have to have somebody working with you who really understands research nutraceuticals. You need we actually work with Chinese herbalists. We actually even formulate some of our own with Chinese herbalists. I'll go with patients to an herbalist and say, okay, this is what I see. And so we marry the best of Western medicine and Eastern medicine, which formulation and delivery is the best of Western medicine. You have to get it where you need it. So, but we're going to reorganize and take down our website for a little while and come up with some very practical things that we can do. We've just not had the time in the past few years fighting the plague of corruption, but and I will answer, and I will give you the names of people, companies, doctors we're trying to build that network as big as possible, and that's my goal of being on here with you, is to find doctors, nutritionists, naturopaths, DOs, chiropractors, find people who see it and who want to help, and we'll direct them where we need to go, and where they need to go, and offer as much help as we possibly can.
Yeah, I mean, being a chiropractor, and I've always, since becoming a chiropractor, I've not taken any medication, and I used to take medication all the time the first 29 years of my life. Yeah. I was on one drug or another and chronically ill, and then I started chiropractic care, and boom, I was like, my life turned around. The chiropractic alone really helped me, but right. When I started going totally organic, right, absolutely, that brought me up a notch. So my question is, for these people that are watching this webinar, I'm sure that they're as frustrated as I am to find someone who really understands this, because even a minor problem is very complicated for treatment. Right. So you take somebody who has chronic fatigue syndrome or cancer, their problem is going to be even more complicated. Mm-hmm. How do they go through the process? Like, if a practitioner is watching this, and I'm still going to suggest they contact you to get more in depth into this, but how do you go through the process of, because for me it always seems better to start with less supplementation, because the body can only handle so much. Do you have a general protocol that somebody could take and
Yeah, we look at the entire body system. You get the patient in there, and in a long talk, you ask about sleep. First thing you ask about is gut. Second thing you ask about is sleep. So we know so much about, a paper just came out yesterday, said if the microbiome of the gut isn't doing well, antiretroviral therapy doesn't work as well in AIDS patients. Oh, got that. A paper came out a couple of weeks ago, said microbiome's no good in ME/CFS. We knew that. We know that. They look like AIDS patients. Those are those pathways I showed you. So you get a detailed history and you find the pathways, because interestingly enough, there's so much redundancy in the NRF2 pathway, in the RNA cell pathway. If it's herpes virus is your issue versus mastocytosis, the mast cell, what's getting dysregulated? You look, is allergy, is sensitivity, you look at the problem and you try to get to the bigger problem, because again, is we use the term in radiation safety throughout my career, ALERA, as low as reasonably achievable. So like, I want supplements, I want bad food, I want everything I do as low as reasonably achievable, because that's the way you're going to keep the toxins out of your world. So I agree with you. We don't use a boatload of supplements. And in fact, that's usually what I have to do. I get a patient's list and I'm like, oh my God, no wonder you're sick, and how do you even do this? You take 3 of these in the morning and 3 of these in the afternoon. It's amazing. But that said, there are things that can help, and you just have to fix the metabolism. You have to fix the pathways, and right now there are about a dozen key pathways, and we know them. And so we can do a couple of simple tests, and we know them because they keep coming up again and again. The B cell disease, the subclass deficiencies, low overall IgG, chronic infections. Is it chronic infection or is it chronic allergy? And then you're looking at mast cells versus NK, T, and B cells. So there's a real difference, and it's just it's talk to the patient. Get the family history, because I just showed you that. That's how we got everything in our studies. We didn't say it's this. If you look under the light for your keys where they're shining, they're not there. We're looking at the system's biology, the whole system, and where is the key thing impacting you now? What's the thing disturbing the quality of your life the most, and how do I fix it? And there's usually a simple answer.
So for me, I'll just use myself as an example. My father died from Lou Gehrig's disease, and he died at the age of 67, and he was gone within 2 years, and he was in phenomenal condition, but I can tell you, he never ate organic, ever.
Right.
Right. But
where did you live?
Outside of Philadelphia.
Okay, good. Industry is a real issue. So the genetic susceptibilities that we know of are in liver detox. SOD catalase is the genetic components. So you want to focus on the liver in that case, because he had a detox issue. I mean, we all have a detox issue, but the faster you can detox, the better off you are. So I'd start with the liver and ALS. So the genetic things tell you something. So for instance, I don't do a lot with the whole 2ME and all the genetic SNPs and things like that. What I look for is patterns. So when you have a problem in the methylation machinery, that can be felt throughout the body. So you can look, if you have those kinds of information, they can tell your chiropractor, your healthcare practitioner a lot. So I just go straight for the liver on that one and look for toxins as well, and make sure those are out of your environment, because then you're less likely to put the genetic pressure on your susceptibilities.
Right. And what's the likelihood that I pass some of this down on to my daughter?
Well, it's no gene. It's not 1-to-1-to-1-to-1. They're complex. Okay. It's no one gene, one disease anymore. It's multiple. So certainly we see, like we heard from that the caller Jamie, that her son never had any of these things. She has multiple. But as one of the doctors said, sitting with an ALS, a young ALS, turned to the mom and said, "It's always your fault." So it's always your fault, Mom. So,
Well,
Sorry.
yeah, my daughters had a diet of, 80 to 90% organic since the day, she was
Good.
breastfed for 2.5 years, but that's always been a huge priority for me.
Mhm. And we know those modulators. There's a good therapeutic called Protandim. Protandim is I don't know the name of the company, but it's a natural product supplement that's formulated from 4 different things, and it was formulated and in fact has been shown to have a lot of benefit in ALS. And so this would be one I would use to prevent. That would be one that I would buy. It's an NRF2 modulator, and I can put you in touch. I don't sell anything, but I can put you in touch with reps from various companies, because of course then you get better prices and all of that stuff. It's no different than, getting you to the right doctor. That this is something I would say, have Protandim in your family, and you yourself as well, because beyond not putting in bad stuff, it's help your liver detox as efficiently as possible, because we can't, as low as reasonably achievable is still some toxin.
Well, what do you think of colonic therapy?
There's value in it for the appropriate population. Yep.
Okay. And for it's hard to ask specific questions for more of these chronic diseases and cancer, because, as you said, the family history is very important, or the history of that person. But take someone who's pretty healthy and they just want to stay healthy. What kind of recommendations do you make for those types of people?
Well, I do what I do. I tell them what I do, which is go back to real food. I cook a lot in a crock pot. I buy organic. I do Tropical Traditions chickens and whatever food they have. I just love it. I buy really grass-fed, non-GMO. I buy liver. I buy calf's liver, because that's where you get so much B vitamins. I do the bone broth. Louise Hay and Heather Dane have a book about the bone broth cure. It's not realistic for me to plant anything. I live in a 700 square foot apartment. But I go to farmers markets. I'll take products as breakfast. So I use a particular vitamin rich juice that has a lot of benefit of oranges and tomatoes, the lycopenes, B vitamins. I take that juice with a supplement in the morning and a hard-boiled egg, cage-free, and that's breakfast. So I don't even know where the grocery store is. My husband goes, but I think every now and again we run out of toilet paper. Once he was out of town, and I didn't know where the grocery store was.
That's so funny.
But I don't go. And so I go back to cooking. I use 20 mule team borax to clean and for my laundry because it's potently anti-inflammatory and it binds up all these toxins. It binds up molds, mycoplasma. I live by the ocean. I can't get rid of the mold. If I look at the glass behind me and I run my finger up it, even if I dusted it yesterday, I don't use, I try just as much as possible. If you can't read the label, don't buy it. Don't eat it. And try not to let your kids eat it, though that's a lot more difficult. But I cook in crock pots. I don't cook in microwaves because they cross-link the nutrition, the DNA, so you can't get the nutrition out of it anyway. I guess there's a lot you can do in your environment. I get real sunshine. I go out every day and I get an hour of sunshine. I don't put anything on my skin but the highest grade coconut oil from glyphosate free. And you'll find if you're getting enough carotenoids out to your skin, you won't burn anyway. You won't get skin cancer anyway. So you'll be protected from the inside out if you eat well. Now, I don't recommend frying out in the sun with coconut oil. I rarely spend more than an hour a day, but I get natural sunshine for vitamin D. Just basically, I just don't put anything in. And so you end up saving money because we don't spend a lot of money in the grocery store. I just won't eat it. And so it doesn't make any difference.
So I have a question from Gloria. She's got two questions. First is, how do we bring this up to a doctor to get them to look into it, to figure out how to treat it?
Well, I think you don't really bring it up from the retrovirus angle. You bring it up from a symptoms angle. You bring it up from an immune system angle. You bring it up from a detox angle. Fasciculations, difficulty sleeping. That's what a doctor can treat. Autism isn't anything a doctor can treat. Encephalopathy, inflammation of the brain, you can treat that. You can stop that. If lymphatics, if edema is the issue, look, I got this edema, I got this swelling, I can't get my rings on, I can't move, I'm in pain. That's inflammation, and you can treat that. And when you start treating systemic inflammation with lymphatics cleanses and constipation is a problem because the toxins sit there. Diarrhea is a problem because something's wrong. I also eat a lot of fermented foods. I'm Hungarian and Austrian. I eat a lot of sauerkraut. I eat really good meat, and I eat meat. We need those proteins. We don't get creatine without meat, So you have to have good all sources of protein. We're not made vegan. And so vegans have different challenges, and they'll have to buy that creatine and those proteins and those building blocks from clean sources if they want to go that way. But there's a lot to it, Gloria, as far as bringing it up to the doctor. You have a chronic disease. You have an acquired, a chronic disease that I don't care what name you name it. It has the problem of it. You can't sleep. You have the gut dysbiosis. You have the debilitating fatigue. You can't stand light. You can't go outside. You can't work more than a few hours a day. The doctor will start investigating that. And again, we don't need to test for the viruses. We know they're there. 15% of our genome is there. Why are they messing up our immune system? What else is going on? And just don't inject anything. The answer is absolutely no. If you are sick, not a vaccine, not ever. If you walk into your doctor's office with a respiratory infection and that nurse says, oh, it's time for your DTaP, you're going to end up in vaccine court with me acting as an expert witness saying, what do you mean you injected a sick person? So, and this is crazy. Vaccines are drugs. Take you just as much as we seriously consider every drug you put in your body, I put in mine. You're going to stay healthier if you do less. Don't even do aspirin if you don't need it. Certainly not Tylenol to mask and create liver function. And of course, we all know how to, not do recreational drugs. But that said, medical marijuana and some of the cannabinoids are very valuable treatments in all of these diseases, and you should seek out a doctor who will listen to you.
Ariane asks, I hope I pronounced her name right, what antiretrovirals are effective against XMRV and MLV related retroviruses?
Just about all of the ones that were discovered for HIV. The best are the Retrovir, AZT. Suramin, though it wasn't an antiretroviral, it's an anti-purine metabolite, it's an anti-parasitic drug. Suramin is very potent. Viriad works very well. Integrase inhibitors, Icentris, Raltegravir, I don't know all their names right now, but one of the things I spend countless hours each week is in the HIV literature, because our government's spending a boatload of money to cure HIV. They talk about cures of HIV, curing 2-year-olds, and then they turn around and tell you these drugs aren't safe. Well, that the endpoint of that meeting in 2000, the first and last XMRV workshop, somebody asked the same question that Ariana's asking, and one of the other longtime AIDS researchers answered that and said, basically, in a patient population where the suffering and loss is so great, the benefits far outweigh the risks of trying these things. And we are trying them, the we being the scientific industry in diseases like multiple sclerosis, Parkinson's disease, ALS. There is a clinical trial of antiretroviral therapy in ALS. The problem with that clinical trial is you have to demonstrate you have a particular kind of HERV, of human endogenous retrovirus. Well, it's no more that than it is an XMRV. And yet, so they biased the study to start with by just not letting all comers with any reverse transcriptase, or which of course we all would have, especially chronically ill. So they find what they want to find, and that's the real plague. But essentially all of the retroviral therapies being used can have benefit in one patient population or another, but some shouldn't be used. You really need somebody who's used to that. That's where Mikovits and Rossetti MAR come in, because we work with doctors who aren't used to treating with those therapies, because we've used them for now more than a combined 75 years. So we know what works in the various neurological diseases. We know what doesn't work, and we can mitigate a lot of the potential damage by choosing properly and getting the nutrition set in the beginning, getting that patient to the point where they will respond. Because what we learned in HIV, and that's why Magic Johnson, if you let them get sick, you can't cure them no matter what. So if they are so far down, the sooner the better. We've seen people ill 25 years get their lives back. So it's not over. It's not without hope. And there's a great deal of optimism that we can do a lot if the medical community and people like you go out and look at these patients and say, well, wait a minute, maybe you're not crazy. Maybe you really are. Maybe you really are sick, but that's the problem. When you say, oh, that's gay man's disease, or gay, I can't think of all the things they used to say in the 80s when I walked out of UVA, that diploma sitting behind me in 1980, and I saw all of these diseases, and you were looking at, it's not so socioeconomic, folks. We're all human, and there are contributing factors that we can find, and retroviruses are clearly one of them, to every chronic disease that I had listed on that slide.
So recently I was interviewing somebody, and the topic of leaky gut came up, and how one of the issues is with glyphosate, that it's causing this leaky gut. And the person I talked to recommended a product called Restore to help basically bring the fibers of the gut back together. And
that's a really good product.
Good. That's right. Now, is that a product you would recommend for anybody, or?
I think where there's a leaky gut, demonstrated leaky gut, I'd start there. Okay. And in fact, the physicians do that. You'll do that before you add the antiretroviral. The antiretroviral will eventually heal the gut and make it non-leaky. But if you start putting a lot of probiotics and try to normalize the good bacteria, then you're going to have them on the wrong side of the fence. Right. And if you're leaky, it means that good things in the wrong places are bad things by definition. Yeah, too much of a good thing is bad. So you don't want to be doing all these digestive enzymes and everything when you've got a leaky gut, because you're probably making the situation worse. So Restore to heal it, and the antiretrovirals will eventually heal it, and then we can put in the strains we know will aid in response to the drugs or the natural products.
Okay. How do you know if you have leaky gut?
If you have GERD, if you have pain, if you have diarrhea or constipation or alternating any one of those things, especially all the reflux. We have a lot of reflux, because you see, leaky gut, the microbiome's not the same from the throat to the anus. The microbiome has different sections. So if you're leaky from here down to here, you have, if you're taking Tums, you've got a problem. if you're taking Tums, don't take Tums. They're loaded with aluminum. Take Restore. But it leaks from different parts from here all the way down. So any kind of problem from top to bottom, any kind of indigestion, any kind of gastrointestinal reflux disease, any family history of IBS, Crohn's disease. I mean, just keep going.
Is there any contraindications?
To Restore?
Yeah.
Not that I'm aware of.
So if someone suspects they have this, they could take it.
Yeah.
Okay.
Just like you can buy Tums over the, even though they're hurting you more than they're helping you, and putting a Band-Aid on a problem isn't the issue. You want to heal it, and Restore will heal it. And that's what you really want to do.
Yeah, I never heard this part until literally last week. Zen Honeycutt brought it up. We were talking about the study she had done looking at glyphosate in all the vaccines. Right. And she did a great job of explaining how glyphosate.
She's fabulous.
Yeah, she did a great job of explaining how glyphosate affects the gut and how you can heal, because she had her own child was very ill. And so I thought that was really fantastic. So I wanted to ask you about that as well. So this question I have is really off topic.
Okay.
I like to throw a curveball at you every now and then.
Sure.
So do you know who Candace Pert is?
I love her, worked with her.
Yes. So Candace.
She died.
Yes, I know.
Normally my last slide is dedicated to her.
Okay. Oh, I didn't know that. So I saw Candace Pert speak back in the early 90s. I had just moved to DC. She was speaking at a small place, and I think it may have been before her book Molecules of Emotion came out.
Correct. And
she talked about basically, as a female in the scientific world, you're pretty persona non grata. And I'm kind of guessing that you had the same experience with that.
Correct.
So that happens pretty much across the board. You don't see a lot of women that are given the same respect that men are given.
Well, absolutely. That's why I showed you Elaine DeFreitas, and the loss of her life. Well, Sidney Grossberg and all those other people that were wrong, and those men, and Bob Gallo. That's why that slide said they're in the National Academy, because that talk was actually, I gave a talk a few years ago about misogyny in science. And, it's like you can bully people because you're a man, and in today's world, there's nothing about, equality, and never was or will be. So Candace, in fact, we developed peptide T, which was what the whole movie, The Dallas Buyers Club, was based on. So we had done those original experiments, and I think I was in my 20s. So Candace did a great deal, to show me how to be really, I was tough just because I'm my mom's daughter, but, she showed me, how to take it. And in fact, she had a poster on her wall and it was in her obituary when she died. It said, if they're running you out of town, get in front and make it look like a parade. With that.
That's so funny.
And so I that was my last slide. I'll put that on one of our talks. Maybe the next time I talk, I'll make that my last talk. But yes, there's no doubt. And in fact, if I didn't work with Frank Rossetti today, Frank Rossetti being the father of human retroviruses, and they went after me. They didn't go after Frank. And so they threatened me through Frank. They said, because I said, no way, not throwing away the data, showing everything. I don't care what you do, do whatever you want, So, they got on the phone with Harold Varmus and Michael Bush and said, fine. Then we've got Harold Varmus on the phone, and you either put your name on that paper right now and say the blood supply is safe, and we made a mistake, folks, or Frank Rossetti and Sandy Rossetti are fired for fraud right now and lose their entire retirement. And it was September of 2011, and they had worked combined more than 80 years. And it's like, and Harold Varmus is on the phone. He's the head of the NCI, the threat was, since I'd already, I was shortly thereafter fired. I was hunted down. I was jailed, for months. So I knew they'd carry out their threat. And so, you know, but they didn't do it to the man. All they did was disgrace him and force him and Sandy into retirement, which is horrific for our you know, using those brains. But the good news is Frank is still here, and he's about 3 doors up the street. And so, so we have to go. I'm going to the office in a few minutes, which is down on the beach, where we sit by a chair and we just talk about these problems and solve the problems and help the patient. So Frank Rossetti's brain won't get out of it until he's 6 feet under, and he's never given up on me, and so it's not a problem, and sooner or later we'll expose him. I mean, you know, what's Lipkin going to do? Sue me? I called you a fraud. I called Tony Fauci a fraud. I got it. And we've got William Thompson and the entire CDC, you know, lying to a federal to a congressman. We've got y'all. You know, why don't they sue me? Why haven't they said one word about this book? Because every word in it is true. And don't get intimidated by how fat it is, because most of them are references. Most of them are references with the quotes, the time, the July 22nd invitation only meeting. You mean, what are we going to do to protect our lab workers? We can't protect our lab workers. Well, in fact, they infected me. So when everybody asks, what do I do to keep myself safe? I'll tell you exactly what I do. I take Protandim. I take a really fine supplement, the juice I mentioned. I eat this food, and only when I get really driven under do I start looking like a multiple sclerosis patient, walking and talking funny with brain fog, because yeah, they infected me, and they know they infected me, and I'm coming after them. And I just keep saying, we're coming after you because we got the truth, and the truth will prevail. Whether it's now or 10 decades from now, I hope it be today, so we can save some from suffering.
So Ariane asks, can she and her husband contact you? They have many questions. They were both recently diagnosed with CFS/ME. They emailed you about 3 months ago. Didn't get an answer.
I thought you already emailed me. Okay, yeah, I'll answer you.
Okay.
Yes, I knew the name, so I will answer. And so yes, people can email. That's why I put that email address on there. I get, if I don't answer you in 10 days, come back to me and say, just a reminder, Dr. J, know you're busy, because what happens is it gets buried in literally thousands.
Right.
And I remember it, but I often can't find it. So we're trying to fix those little problems by, we've got an office here, a new office, and we're going to reorganize, and we've got a phone that's not my cell phone, so that we can leave messages, and then we can get to people and schedule appointments and talk and get those data where we need it to be in various secure drop boxes and things like that, because we are doing as much as we can, and I do remember you. So do email me again, because I try to answer as many as I can, and if not, direct you to a doctor. Almost always my first question is, where are you? Because wherever you are located, whichever state, if you are in California, my life is easier because I can get you more therapies and more doctors. If you're in Indiana, my life's really hard because it's owned by big pharma. But at any rate, we're learning, we're working together. Doctors like you, Nancy, around the world, doctors who will work with us, and I can simply direct you to a doctor, and we can all get on a Skype and do it that way.
Well, it may be good for some reasons to live in California, but for other reasons, it's not so great with what's going on with the vaccine mandates. And
that's why I will have missed that, because starting in August, my mailbox was, help me, help me, help me. My kids aren't vaccinated and they can't go to school. I've even got people asking us to teach their kids. So I said we would, because it would be a great deal of fun. Frank Rossetti knows Shakespeare better than Shakespeare. So we could teach a few things and have a little bit of fun. And it's a nice setting here, but obviously that's not gonna solve the problem. But living in California, for that barrage of those mandates, I'm spending at least 30% of my time on trying to stop that right now.
I don't know if this is a possibility. This is my last question for you, but I'm going to throw this out there. There's a lot of people who have these illnesses that do their own research, that for whatever reason, they don't trust medical doctors. how would you feel about doing a series of, self-help workshops that were really designed for people? We could take, this health problem, in one hour workshop. These are the steps that you take to start healing. These are the kind of, like, I know that you can find a way to test like vitamin D. You don't need a doctor to do that. There's plenty of labs that you can actually do that. Would you be willing to do something like that? And for anybody who's listening, of course, there will be a fee charged. We've been doing these for free, but it's not practical anymore to continue to, we'd have to charge some sort of nominal fee. Are you comfortable with doing that?
Sure. I've practiced without a license most of my life. So that's what Frank used to say, Judy, whenever I brought him one of, I'm the person that brings stray pets in off the street. Frank, it's just this cancer. It's just this. We've been doing it since 1980. No, you got to see this. No, you got to see this. And that's how I got myself in all this trouble with ME, of course, as I looked at the patients and I looked at an outbreak and then said to Frank, and he said, oh, that's ridiculous. They'd be AIDS patients. And I said, yeah, that's what I was thinking. That's in the book. So that was, a decade ago now. But sure, I'm willing to do that because we're thinking about, how better to help. And I appreciate patients who do their own research. I just caution that you're going to do better research in books. Go get Suzanne Humphrey's book, Dissolving Illusions. Go get James Lyons Weller's book, Cures versus Profits. When you get Plague, and it reads every chapter at a time. You don't have to read about the jail chapters or things like that. If you're only interested in the fact that you were a lab worker and you were working on your PhD in some mouse studies, and all of a sudden you got really sick and you never recovered. So many of those lab workers come to me these days by email. And so we can develop teams of people, and they do their own research and they're very good, but there's a limit to the research because the research is what they want you to see. And so it doesn't get published. Now we have the internet, so we have a lot of advantage with the internet because the guardians of what gets published, the censorship of literature, the only place that still remains is in vaccine research. And even now with books like the autoimmunity book by Yehuda Schoenfeld and Lucija Tomlovic, those books, that autoimmunity book, I'll make sure we get all of these books on our website because they tell you the story. They tell you the dangers. They tell you the fallacies behind herd immunity and things like that. And these doctors, that we can build a bigger network and doctors who will help around the world. So yeah, do your own research, but don't forget the books. I know people don't read books anymore, but that's why we wrote them, because you can't ever make it go away. But
I think starting with like OPQRST, like how to figure out when it started, what makes it better. That could be the first one. And then the second one could be a specific health problem. So everybody is responding very positively to the idea of doing this. They're all happy to pay as long as it's affordable, which of course it will be. But, we feel that, I feel that, because a lot of people that are watching this are lay people as well, I know for me, I like to, I know myself better than anybody, and I know I have a total trust in natural things, but finding the right products is essential, especially with what, I don't remember I think it was Stephanie Seneff was saying that glyphosate is even in gel caps. So that you have to make sure you have the highest quality product out there.
And of course, then those get expensive. And so you have to go to companies who test for those. And then people get mad at me. And often I'm just recommending the products that are, and yes, they're expensive because they do that. And you don't, like even gel caps. So people will package, and even the herbs, our herbalist will package her own, but she knows where to get the gel caps that are not. And so I'll send you to that herbalist because I trust her, or we can have that herbalist come on and do a webinar. I think the first thing we have to do is how to take a good family history.
Right.
because that's what everybody needs to know, how to ask those questions, because the key to every study that we did well and every little bit of investigative work, when I look at these vaccine injuries, which aren't easy, they're not easy because every deck is stacked against you and so much else happened in these people's lives. But often you'll find something subtle like, oh, they're HLA-DQ-V1, and that's associated with Behçet's disease and all these autoimmune diseases. So you start out with, where's that susceptibility? So if you work as in a coal mine, if your grandfather worked in a coal mine, if you're organophosphates, so how did the environment matters? If you work by a lot of, if you're in the IT industry, if you're working with the internet and all of this stuff, you're getting a lot of the electronic magnetic frequencies dysregulating your immune system. It's a whole lot more than just intake in that environment. So that's where you need to that the first one will be, okay, everybody, and then everybody will do it together, and we'll do a few key examples where we learn something that we wouldn't have known if you didn't ask the right questions.
And it might be good to use somebody who's on the webinar today as we'll have that person do their case history.
Yeah,
so
if Ariana or Gloria want my help, or Jamie with the family, we can already see there's a couple there.
Yeah.
Happy to work with them as we move forward.
Okay, so I need the people that are on today to send me an email, embodychiropractic@gmail.com. That's MBODY chiropractic@gmail.com. And let me know if you're interested. And then the next thing, of course, is finding a good time. But I'm thinking a series of 5 or 6 or 8. Yeah, I mean, let's come up with
find a time where people can do it, like Saturdays, Fridays after work, whatever that works for me. I'm okay most times.
And we'll come up with a syllabus. And the thing is again, they'll be recorded. So that anybody could, if they need to, they can't come online, they could still, they could still be part of it. If they want my email address, I'm going to type it in at gmail.com. There it is. So yes, family history is important. But again, are you next to a power line? There's so many pieces of the puzzle. And I wanted to just mention that before we get off, is that I kind of new to working with kids with autism. I've been working with them for a long time in dribs and drabs, but just in the last year I've had an opportunity to meet so many more, and every case is different.
Correct. Absolutely.
Every case is different. And the thing is I would have people come into my office, because I've worked a lot with bowel issues, that every case, you have someone with constipation and there could be 10 different things that actually help them get better. Right. Same thing with back pain. I mean, just back pain. You could have back pain and there's 10 different things. So history is so important to really understand.
I hate labels. I just hate labels. That's why when you call it ME/CFS, that chronic fatigue, well, every single disease has profound fatigue. That used to be one of the warning signs of cancer. If you're unusually tired, if you gain weight suddenly or lose weight suddenly, that's half of the people that we work with. Yeah. That's all of ME. That was cancer. You have cancer. So anyway.
So I think it'd be really great to also take this to another level, which is not only talk about what can be done, on a nutritional level, but also this idea of maybe even following this person through, a period of 8 or 10 weeks, and see what kind of changes they get.
Oh, it takes time. It takes time.
Yeah, I mean, they might be subtle, but they're starting to see consistently that their energy's improved or they're digesting their food better. But anyway, thank you guys for coming on, and everybody is really interested in partaking in this, because, people would like to have a little bit more independence and, be able to.
I mean, we have to go back to old-fashioned medicine. We didn't go anywhere. Mom's like, should have thought of that when you cut your finger off.
Right.
Thought about it before you put it in the food processor. And I'm like, oh, come on, Mom, can't we get some stitches? Nope. But now we go back. Now we go to doctor for wellness visits, which is precisely where you'll get sick. And and we're well. A ruler on the refrigerator. Everything has to go back to a simpler time.
Yeah, and my daughter, she's now 12 and a half.
Mm-hmm.
She was going for well baby visits during the first 11 months of her life, just because I wanted to see how big she was and how tall she was or long she was. She wasn't getting vaccinated. And since then, she's only been to a medical doctor once. That's it. She gets chiropractic. We eat organic. Right. I treat her with food. She's had acupuncture, homeopathy. That's it.
Yeah. My grandmother died when she was 95 years old, and that was the first time she was in a hospital, and she got pneumonia because they took away her champagne. She lived on full-fat ice cream and champagne, and somebody decided that they'd take away the champagne. Well, the fermentation process, she changed her microbiome, and she got an infection, and she died. It's like, come on, folks.
Yeah. And, the other thing is I have a good friend of mine who's pretty much an expert in, she's a cook, a chef. She could come in and do one on, making your own ferments and bone broth. Oh, yeah. And
you have Heather Dane. I can give you some names of people who will go on who I've done shows with here in Carlsbad that are talking, and they do the cooking classes. So I'll get to Heather and find out all the wonderful people I've been meeting and get their last names and their shows, and we'll get them on there, and we'll in fact, they have a radio station, so we could get up there right up at the top of the hill, and three of us can be in the same room talking at the same time. It's fine.
Fantastic. I love it. Yeah. All right. Thank you all for joining us, and we'll see you soon.